And in today's class, we will cover types of biological networks. So 3 main types: metabolic network, protein signaling, and gene regulatory networks. So they all have their challenges, and we will learn how to abstract them using ordinary differential equations. And if you guys are not familiar with the ordinary differential equations, I'll give you an overview to do the exercises today. So today is more like a first part of the class. I will go over the slides, and then we'll do some exercises and discussions. So we'll do the representation of these networks using ordinary differential equations, and also in the code. So we will build the very simple Tellurium code to run them and see what they're doing. So let's get started. So this is the outline of the class. That's all okay. Recommended reading if you want to go more in depth about it. So last time we covered the types of networks in general. So in economy, in AI, so on and so forth. Today we'll go over the gene regulatory network, metabolic network, signaling, protein signaling networks, etc. So what comprises a network is topology, the how many nodes you have, how they're linked, and their internal states. And in last class, we quickly discussed what is a model. So what is a model? James presented this slide. I think it agrees more with what you were saying. So you have this model, this black box. You give it an input. James also would say you also have to provide a structure for this model. And then it gives you metrics, outputs. And these metrics can, if they are good enough, if the model is validated, they are making predictions about the system. This is my slide about model. And what I think model is a representation of reality. And I like to use this figure on the bottom right. So the way we do summation, we have 2 things. We join another thing, and then we have 3 things. We are abstracting the things using numbers, and the operations are an abstraction of what we're doing with these things, and the resulting number is an abstraction of the result. So the numbers themselves are representing something. They are abstracting something, and we are using those numbers and operations to represent what we are doing with these things. From a Google search, you'll get imitation as a model. I think it's already a good enough, like a very short and very good definition of model, imitation. So there is something out there, and we are trying to imitate it in a more simple way. Some people use maps as example of model, like real maps, like cartography charts. And one example is, the top right, this map, let's say, of a building. It's not the building itself, but it's representing it. And you can even build these kinds of models to study, for example, mechanical tension on the building. Last time we also discussed dynamics. So dynamics on the network it means that the state of the nodes are changing, and the dynamics of the networks is how the networks are changing, which means the topology, how the nodes are linked. Okay, all good. Next. So we saw that chemical reactions can be represented as networks, and we can represent these chemical networks as mathematical models, such as ordinary differential equations. So are you all familiar with these things here? You're familiar? What about you, Niraj?
Yeah.
Okay. So what is happening here is that you have A combining with B going to C, and a rate of how fast it happens. So you can write the rate of the rate of change of each node A, B, and C, and it depends on the state of the nodes. And if you consider the law of mass action, the rate is proportional to both concentration of A and B. And do you guys know what is the idea behind the law of mass action? Why is it proportional to A and B? You're making a reaction. So you're mixing reactant A with reactant B, where the value of A is the concentration of the reactant A, and the value of B is the concentration of the reactant B. And you're saying the rate at which I lose A and B is proportional to the product of A with B. Why? Why A times B and not A plus B or A squared times B or exponential of A and B? So I'll
Yes.
make a very simple scheme drawing here. So let's say my reaction is happening in a lattice. So here I have my beautiful lattice, and I have a certain concentration of A. So let's say I have one atom or molecule of A for this space, and it's going to be randomly located in whatever of these lattices. So this is my molecule A, and this is my molecule B. It's a circle. Okay. Now, what is the probability that they will be in the same spot at the same time to react, forming C? It's the number of molecules I have of this one times the number of molecules I have of this one. Of course, the number of molecules divided by the number of lattices I have, which is the concentration. So it's whatever the probability of this guy being here and this guy being here, right? So it's the probability of this happening, right? So it's the probability of the one being there times the probability of the other one being there. That's why it's a product. And the more of these things I have, the higher the probability of them coinciding in a certain place. So that's why we assume that the rate of change is proportional to both of the concentrations, and the probability of them interacting is the product of them being in a certain position at the same time. So this is, of course, a first approximation. It can be more complicated if you have a very, very high concentration. But for low concentrations, that's valid. Besides simply flow of mass, so here we have A plus B going to C, we can also have some species S1 converting to species S2, and we have another thing called a catalyst, this A node, and the A node is promoting the transition from S1 to S2. So what is a catalyst? A catalyst is something that participates in a reaction, but it doesn't get consumed by the reaction. It is recycled. And what a catalyst is doing is basically lowering the activation energy Let me hide this thing here. How do I do that? Hide. Oh, perfect. That's much better. So it is lowering the activation energy of the reaction, so it has a higher probability of happening, or of course, in large systems, a larger probability converts to larger rate of reaction. And in this left figure, for example, have an enzyme with an activation site, and when two products combine at this reaction site, the reaction happens. So the enzyme is basically promoting the combination of these two molecules. In that case, the molecules start linked, and then they go to the enzyme. The enzyme breaks the bond between the two. So there are many forms of enzymes out there. We also saw how to represent them in a simple Tellurium code. So the advantage here of Tellurium, of course, is that you write the reaction as they look in the paper. So A plus B and then right arrow C, very simple stuff, and the rate of reaction. Sources for network structures, and we'll start with metabolic networks. So when you see at this slide, you see this huge network, which we'll try to memorize until the end of the course. This is one of four, so we'll memorize all four of these networks. No, I'm joking. This is to show you guys how complicated it can be. So this is basically the Krebs cycle. It's how cells produce energy. But this looks much nicer. So we have a pyruvic acid comes from glycolysis, and pyruvic acid converts to acetyl-CoA. I think pyruvic acid is the one that enters mitochondria, and all that stuff, the cycle happens inside the mitochondria. This is super, super fast. It's microseconds to seconds in a single cell. Of course, for the entire body, it's going to take minutes to hours, like all cells, when you start eating something, etc. And the keywords for metabolic networks is energy. You have flow of energy. It's as if you're burning the sugar, let's say. Sugar is flammable, okay? You can set sugar on fire. So your body is doing that, but in a slower way and controlled way. So instead of going straight from sugar to CO2 plus energy, you are going step by step. First, sugar becomes pyruvic acid, then acetyl-CoA, then citric acid, and it's going down a staircase until you release the CO2 and release energy in the form of ATP. So it's kind of a controlled and slow combustion of sugar. And why would you work with metabolic networks? So the scientific or the research interest in this is, for example, learning how the environment affects it, how the substrate availability affects it, and of course the pathway structure. If you add a component, let's say you add fat to the Krebs cycle, what happens? Or you take out fat, what happens? It definitely behaves different. There is a proper notation for these metabolic networks. The notation you can see in this website, and it helps standardizing these networks. So sometimes if you see this one, it's not so clear what the arrows mean. Here they probably mean reactions and flow of mass. But sometimes you are ignoring flow of mass, so you would use other types of arrows, or for example, catalysts will use other types of arrows. Now, protein signaling. Protein signaling, oh, I forgot to mention that besides flow of energy, the metabolic networks are also building cell components. So lipids, proteins, so on and so forth. In the protein signaling networks, here we are talking about communication. So how do cells communicate spatially? How does a cell talk to itself? So, or the entire body. So the autocrine signaling is when a cell releases a protein that the cell itself senses. So it's basically sending a message to itself. Intracrine is when the same thing is happening, but you don't need to release the molecule. It's a protein inside the cell telling the cell to do something. Juxtacrine is when two cells in touch with each other transmit signals across their cell membranes. Paracrine is when one cell releases a molecule and another cell in the vicinity senses this molecule. And endocrine is when a cell releases substances into the bloodstream and it can reach very distant cells in other parts of the body. And the ways at which proteins can communicate, can pass information to other cells and inside the cell is by some reactions. So for example, phosphorylation is a reaction that changes the state of a protein. And once you phosphorylate a protein, it changes its structure, it changes its shape. And now it can behave in another way. So the classes of these proteins that contribute to these communication process are kinases, which are proteins that add a phosphate group to another protein. A phosphatase, which is a protein that removes a phosphate group. GTPases, receptors in cell membranes, and ubiquitin ligases. The representation of these networks involve first flow of mass. So S1 going to S2, it means you are losing S1, gaining S2, and A promoting the transition. That's a catalyst. That's what we saw before. But now we have another type of arrow, this blunt arrow type, are inhibiting the transition from S1 and S2. So imagine you have a reaction happening, and then a new protein is produced in the cell, and this protein is binding to the reactant S1, and now the reactant S1 is stabilized, or it is prevented from transforming, from becoming S2. So there are many ways these inhibitor can act, and basically what is doing is it is slowing down or shutting down the reaction. So a promotion, so the upper charts, they are saying that the rate at which S1 is being converted to S2 is it grows with the concentration of A. So that's the catalyst. The more catalyst you have, the faster the reaction happens. It can be represented as A is promoting S2, or the more formal way, more complete way, S1 becomes S2 and A is promoting the transition. On the other hand, the inhibition, your rate is lower, it's lower when you increase the concentration of your inhibitor R. So R is slowing down the reaction. Again, two ways of representing R inhibiting S2, which means R2 is being produced less, or the more formal way, something is becoming S2, and then R is slowing it down. These are all the representations. So sometimes we're going to, I don't know, a certain website, Wikipedia, whatever, and you see A, then these ball arrow, this circle arrow, going to S2, you have to interpret that as something is becoming S2 and A is promoting the transition. So when it doesn't specify what is becoming S2, so S1 going to S2, here we are not caring about mass conservation. S2 is coming out of nowhere, nothing is going to S2, and A is promoting that. So because biology is too complicated, sometimes we don't care about mass conservation. So you have, let's say you have production of protein, but when you produce a protein, you release water. Okay, but water, we don't care about water in biology because it's everywhere. The body immediately handles that. So if you have more or less water, really you don't care. So in certain reactions, you ignore certain reactants, you ignore certain products. So you can write something very simple as nothing goes to S2 and A is promoting that. In the bottom here, we have the inhibition representation. And same way, if you see these R inhibiting S2, it's as if nothing is generating S2, R is inhibiting the process. Or if you care about mass conservation, then S1 is becoming S2 and R is inhibiting the process. So some mechanisms of signaling networks are complex formation. So you have two proteins, they attach, forming another protein. So that's the case, for example, ribosomes. Ribosomes have two parts. One part connects to the RNA, and the other parts go over it and encloses the RNA, and each part has different functions. Phosphorylation, it's also talked about as activation of a protein, is when a phosphate group is added to a protein via a kinase. So the kinase is this enzyme, this extra protein that brings the phosphate group to another protein. The phosphatase is the inverse. So the first one is the phosphorylation. You add the phosphate group. The dephosphorylation when you subtract that phosphate group. There is also ubiquitination of proteins. So ubiquitination of proteins, you generally require a ubiquitin ligase, another enzyme, another catalyst that will bring the ubiquitin, it's another protein, and it does that over and over, adding multiple ubiquitins to the protein. And when the protein have a certain amount of ubiquitins, it's a protein targeted for degradation. So that protein becomes becomes basically trash inside the cell. It's going to be degraded in the proteasome. So some questions concerning protein signaling networks are, for example, and did I talk about everything here? I think I missed something. Receptors. Yeah, I talked about that. Some typical questions about the protein signaling networks is what happens when a certain protein is present or not. So let's say your cell is responding to an external signal. If that signal increases, what does that cell do? Does that cell undergoes apoptosis? Does it change its behavior somehow? Does it change the cell ability to migrate? So for example, cytokines, when you exercise or when you hurt yourself, the cells start releasing cytokines, and the cytokines promote migration of, for example, macrophages, neutrophils, and those defense cells will help first handling bacteria, but also regenerating the tissue. So when you have higher concentration of those cytokines, you are increasing the migration capacity of those lymphocytes. And so far, we haven't talked about spatial structure. So we are saying that all those proteins, all those molecules are everywhere at the same time, and they are all interacting always. They don't have to travel from one part to another, which is not realistic. So a protein comes from an RNA that came from inside the nucleus. So it has to travel from inside the nucleus to outside. And some proteins, for example, if the cell is doing endocytosis, and I'm using lots of bio jargon. So if you are not familiar, please stop me and, oh, what is this? What is that? Okay, feel free to ask me, for example, what is endocytosis? Endocytosis is when a cell eats something from the outside. So the cell membrane here, that protein, that stuff binds to the cell membrane, and the cell membrane envelops it and brings it to the inside. So you have motion inside the cell, and these proteins, they are diffusing and being transported across the cell. And these means that our assumption that all these reactants are everywhere at the same time is not entirely true. So we'll keep with this assumption, but it's good to keep in mind that some processes, for example, here I'm showing cell migration. In cell migration, it is critical that you have spatial heterogeneity. So here there is some signaling protein in red. That's probably the RAC GTPase, and the RAC GTPase promotes actin polymerization, and the cell migrates. If you don't have this, if you assume everything is homogeneous, you will not have migration at all. These systems are modeled not simply by networks, they're modeled by reaction diffusion equations, which is something a little bit more complicated. I should say a lot more complicated than the ordinary differential equations, which we will work on this course with. Now, to jump to the gene regulatory network, we'll talk a little bit about some basic biology again. So if you're not familiar with something I'm saying, please feel free to ask me, to interrupt me. So what is the central dogma of biology? The central dogma of biology is basically telling us how from the genome of the cell, you get the proteins. So how does a cell know, or where does the information, how the information from the genome becomes the phenotype of the cell? How much of a certain protein the cell has, so on and so forth. So there are two steps here. So first you have the genome. The genome is something you have to conserve. It has to be protected. So you have a transcription step, which is making a copy of that DNA. Imagine you have you are in the medieval ages and you have the sacred book, and the sacred book has to be conserved. Nobody can hurt that book. So you have someone copying the book to distribute the copies, not the book itself. The book, the sacred book, is the genome, and you are distributing the copies. Once the people have the copies, they do what the copy says. And that step is the translation. The translation is from the recipe, which is the RNA, you produce the protein. So the way I memorize it is, so both DNA and RNA are composed of nucleotides. So you have a language, the language of nucleotides. And then you are rewriting the DNA in the form of RNA, but it's still the same language, it's nucleotides. But from RNA, which is nucleotides, to proteins, which is amino acids, you have two different languages. So you have nucleotides in RNA and amino acids in protein. So you have to translate from the nucleotides language to the amino acid language. So that's how I memorize the words translation and transcription. So transcription is rewriting the DNA in the form of RNA to distribute those copies, those recipes, and the protein is produced from the RNA. It's a different language, so it's a translation. It's a super complicated process. It's not as simple as this. And why is it called central dogma? What is a dogma? Dogma is something you cannot question. And as something you cannot question, it's not scientific, but it has this name because in every living being you have that happening. Every living being. If you consider a virus a living being, then this is not a dogma anymore because viruses don't do transcription translation on their own. So if you agree that this is the central dogma, so living things do this, then viruses, for example, are not living beings. So very interesting discussions, but that's not central to the class. So let's keep going. Transcription and translation are super complicated. And just to name a few things, you have components that polymerize the DNA. For example, the RNA polymerase, transcription components, primers, bases, ATP, so on and so forth. And in the translation phase, you have the amino acids, ATP, mRNA, the ribosome. The ribosome is something that we still don't understand exactly how it happens and how it's formed. And it's this fascinating mixture of the ribosome is a mixture of RNA with protein, and it scans the RNA generating the proteins. And multiple RNAs can translate the same RNA strand, and this is a fascinating picture here. So these are multiple ribosomes. They are forming this spiral, and they aggregate like this because they are all translating the same RNA molecule. So this is something else. Finally, knowing a little bit about translation and transcription, we arrived at our final network type, the gene regulatory networks. So the gene regulatory networks, they are controlling the type of the cell. The type of the cell is not exactly the behavior of the cell, how it behaves, because the cell can behave differently according to multiple signals, but it's the type of the cell. It's something that persists more than a quick perturbation. So for example, a neuron is also a human cell, such as a muscle cell, but there are different types because some genes are active and other genes are not active, and it makes the cell be a certain way. So in this example, you have either high or low density of cells, and the density of cells make the cells communicate. So if a cell is sensing lots of neighbors on contact with it, some signals will be transduced internally to the cell, and then you can activate or inactivate certain genes, and these change in the cell transcription profile changes the type of the cell, and this persists for this can persist for hours to days. So one good example of that is, for example, how long does it take for your skin to fully recover from an injury? So you injure yourself, you have the cicatrization. Is that the correct name? I don't know. So you have the wound closure, and then you can see it's a different type of cell. It has a different color, it has a different texture, it has like different properties, and it takes days and days for those cell types to become real epithelial cells. And this happens, it has such a long time frame for this complete tissue repair, because it is a gene regulatory thing that is going on across these cells. So why do we bother with those? Well, so here are some questions that you can use those gene regulatory networks to answer. For example, if I knock out a certain gene, what's going to happen with the cell? If I treat the cell with a certain drug, how is it going to change? And is that drug affecting transcription of a certain part of the genome? So this is the notation of the gene regulatory networks. So you have a node representing the gene, and then you have an input, I, or inducer. And this inducer is making the gene produce the mRNA. So this is the transcription part. And then the mRNA is producing the P protein, right? So this we have here we have our central dogma, the transcription and the translation. You can shorten it. Sometimes you don't care about the mRNA, and then you simply say I is inducing the production of P. And down here, we have a real case of a gene regulatory network. You can see those multiple inputs. And sometimes one of the outputs is an input to the gene itself. And some inputs are promoting the gene, others are inhibiting the gene. So when you have, for example, in this case here, two inputs promoting the gene and one input inhibiting the gene, how do you solve that? Is the inhibitory signal stronger than the two promoting signals? Are the two combined stronger, but each of them are not? So see that there are many concerns when you build those networks. How do you solve these multiple signals arriving at the same node? If you consider that they have to sum each other, or if they have to multiply each other, if they have to be there at the same time, you need both for something to happen, so on and so forth. It's often appropriate to treat these genes as Boolean networks. So you asked me before about the Boolean networks. It's either low or high. It's interesting to use that for genes because a gene often is either active or inactive. So sometimes, and it speeds up the simulation so much to treat these nodes as Boolean. When reading papers, etc., those terms will come up. The first assignment is reviewing one of these networks. So they will use these terms. But of course, I mean, you can always ask us and, meet with us a certain time to, I don't understand at all. What is going on? What does this say, etc. We can help. All right, next. It's very complex. So solving these signals, what they do. And this is our notation. So the simplest notation you can see is P1 is promoting P2. A certain protein P1 is promoting the production of another protein P2. But explicitly writing it as a regulatory network, you write it using the notation, the gene notation. So you have these horizontal bar, then P1 goes to this horizontal bar representing the gene, and then another protein, P2, is coming out of that, being upregulated by the gene. Now, using our previous notations, how do you write that? So it's exactly the same as we were doing before with the protein signaling networks. You have a protein P2 coming out of nowhere, so nothing going to P2, and you have P1 as a catalyst. P1 is promoting the production of P2. You can make it a little bit more complex. For example, P1 activates the gene produces the protein, and the protein decays. The protein is getting destroyed. That's more realistic of our cells. Every protein has a certain phosphorylation rate, and the phosphorylation makes the protein more susceptible to degradation. So how would you do it? You would first consider that nothing is producing P2, and P1 is promoting it. P1 is the catalyst, and then P2 is going to nothing. So you are losing P2. You don't care about what is being generated after the destruction of P2, because that would be unnecessary complexity. So you just say you're losing P2.
So P1 and P2 both are protein. So P1 would also be degrading at this moment, right?
Oh, good question. So Niraj asked, P1 is also a protein, so shouldn't it be degrading too? And yeah, it should. But sometimes you're building your network in such a way that P1 is a signal, is an external signal. So you care about a certain level of P1 and what is the result of fixing that P1 level at a certain value. So sometimes you always want to keep it as simple as possible. But in the case of P2, because P2 is being constantly produced, if it never decays, if it never gets destroyed, in the long term, you'll have an infinite amount of P2 by construction. So P2 will go to infinity as time goes to infinity. So sometimes you put on these decays to stabilize your system. In the case of P1 is an input, so you don't really care much about the decay of P1. However, if you have, for example, P1 being the resulting protein of another gene in your model, then you would care more about the decay of P1, because then P1 could also go to infinity, and then you say, okay, so P1 also decays, so on and so forth. As a note here, the gene activation or inactivation tends to be a Boolean thing. It's either on or off. While mRNA tends to be something more graded. So because you can have multiple mRNA molecules in the cell, and they all translate the protein, so you can have different levels of protein translation, protein production, depending on your mRNA. But in the gene case, it's either on or off in most cases. So the Boolean approach is more suitable to the gene, while the more continuous approach is more suitable for the translation part. So if you consider the presence of mRNA, then you have P1 is promoting the gene. The gene is producing mRNA. mRNA decays. And based on the level of mRNA you have, the mRNA is producing the protein P2. And the protein P2 is decaying. So see, for just one gene, we already have how many nodes here? We have 4 nodes, or 3 nodes at least. And we have one equation for production of mRNA, one equation for destruction of mRNA, and two more for P2. So we already have 4 reactions going on in only one gene. So it can spiral out of control. When you have a huge gene regulatory network, it's often unfeasible to treat it humanly, like you yourself drawing it and coding everything. So some people work with developing the systems that build these networks for you. And this is a very challenging problem, where you build a software that you give the diagram and it models everything for you automatically. Because once you know how to do one, you know how to do the others. So it becomes a very repetitive and boring and high rate of mistakes to implementing these in a model, in the code. Those gene regulatory networks have motifs. Motifs are some basic, simple network structures that when you understand them very well, you are prepared to understand almost everything else. So if you understand a basic gene activation, how it works, a basic gene repression, multiple repressions, a gene cascade, and autoregulation, when you understand how these small systems behave, it helps you to understand how a larger network behaves. So we have our first exercise here, which is representing these guys as a reaction, if they are not yet well represented as reactions, ODEs, and finally as a Tellurium code. So your rate is wrong. Your rate, assuming mass action, your rate would be K times A. Because you are not reacting A with B. You simply have A converting to B. So the more A you have, the more A you lose. You don't need a B, you know? A is not reacting with B. B is just a product of A. A is the only reactant. Make sense? Good. Yeah, so he had this before, so incorrect. It's just K times A. Perfect. So good. That one was fine. I will do the one below. So the one below is A plus C goes to B, right? This one here. And how do I write the ODs? Now I have, sorry, wrong D. dA dt is minus, and then I have a K, and it's proportional to A and C because they are reacting to form B. So this is assuming mass action kinetics. What do I have for C? It's the same. I'm losing C in the same pace as I lose A because they both are required to generating B. So I lose them at the same pace. So I have dC dt is minus K AC. And for B, I have dB dt is the same thing, but it's positive sign because I am gaining matter. I'm gaining mass on B. All good? Let's try the one below. A promotes B.
In the second equation, C is a catalyst, right?
Sorry, C is what?
C is the catalyst.
No, this is no.
No.
So in the diagram, we see that the arrow from C is merging with the arrow from A. So this is denoting flow of mass. So you have mass from C and mass from C is converting to B, because this notation with the arrow is denoting flow of mass. In the case
Okay.
below, where you have this ball arrow, these denotes that A is a catalyst. So you
Okay.
don't lose A in the reaction. All right, Niraj?
Yeah, thanks.
Okay.
Got it.
Good point. It will depend on the activation rate, the activation energy. Yeah, but we are abstracting this change in activation energy by saying that the more A you have, the better the reaction is. So we are simplifying it a lot. So the suggestion from Jen is that the rate at which you produce B is proportional to the amount of A you have. Do you agree with this attempt?
Yeah.
Yeah? All right.
Yes.
There is a more sophisticated attempt. Which is, so this is the first approximation, which is correct. But in biology, sometimes you have a maximum amount of catalyst that will do anything, and above that you have no more gains. So it's a saturated promotion, let's say, a saturation of the catalyst. So imagine, let me think of a real life example. I can't think of a real life example. So let's go without the example and let's jump straight to the solution. So one way people do that is, instead of saying it's directly proportional to A, people use some kind of sigmoidal function of A. What is a sigmoidal function? You are not familiar? You are too familiar because of the neural networks. It's something that goes from one place to the other like this, right? Depending on A. So that's a sigmoidal function. It is bounded from below and from above. It's a bounded function. And it transitions from the lower value to the upper value, or the other way around, nobody cares. And exactly how it transitions from the lower to the upper value, we also don't care too much. Sometimes we do, but the soul of the thing is that you have a saturation. So if you keep adding more and more A, you don't speed up the reaction anymore. One very simple sigmoidal function that, instead of writing sig A, is, you can use this guy here. Sig A is A over 1 plus A. Does that make sense? What is the behavior of this function? If A is 0, you have 0, right? If A is 1, you have 1/2. So, and if A is infinite, infinite, you have 1, right? Niraj, do you agree that if A goes to infinity, this fraction goes to 1?
Yes.
All right. So these will produce a graph like this, going from 0 to 1. What is the inverse of that? I mean, not the mathematical inverse, but the inverse concept. So you have something going from low to high, from 0 to 1. Now I want something going from 1 to 0, because eventually we'll have to model the inhibitor. This is the activator. right? This is the catalyst. Eventually, we'll model the inhibitor. What is a possible sigmoidal function for an inhibitor? Something that goes from 1 to 0. 1 minus this. Perfect. So Jen suggests that our inhibitory function of A is basically 1 minus our sigmoidal function of A. And if you do that, and you do the algebra, you get 1 over 1 plus A. So if A is 0, perfect, Niraj. Niraj wrote before me. So if you have A equals 0, this is 1. If you have A going to infinity, this is 0. So we can write our equation finally as K times A over 1 plus A. This is our very, 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 very simple sigmoidal function. There are others, okay? And for some systems, it is critical that this function is not as simple as it is now. So we'll get to that eventually. Okay. Let's work on this one. C is promoting A going to B. Let's do that. So he's doing, I'll change the notation. My derivative is becoming a prime now. So A prime is minus A. There's a K here. And then a sigmoidal function of C. So Niraj's dBdt is K A C over 1 plus C, correct. And then B is the same thing, but with a plus sign. Perfect, you both got it right. Now, the one below, A goes to B, but B goes to A. Niraj said 1 over 1 plus C times A times K for dBdt, and then with a minus sign for dAdt. All good, correct. Now, I will go to the Tellurium thing, and I will implement one of them. So which one do you guys want me to do to see what happens? First of all, I have to... Where's the... Yeah, eventually... Yeah, here. And... Yeah, this one. So let's maybe try... Let's try what Jen did in his attempt. So his first attempt was the correct attempt, which is A goes to B at a rate K1 times A. Yeah. And then you also have B goes to A. Hopefully that works. At a rate K2 times B. You can also simply say K1 times A minus K2 times B and not have a second equation. Both work. And here we will start with A equals 5 and B equals 5. And K1 equals 1. K2 actually equals 1, 2, but actually let's do A starts at 1, B starts at 0. And let's run this. TE is not, of course. So this downloaded, hopefully, so I can import it. Okay. By the way, this is in Colab. Have you seen this? No? Okay, so here what I'm doing, and you will probably have to do that today. I am installing Tellurium in Colab. So you will create one of these sheets for yourself, each of you, and I will copy and paste these snippets in the chat to facilitate your life. So this first snippet is basically installing installing Tellurium in your Colab, and then down here importing all the good stuff. And then let's see if it runs now. No, it doesn't. Oh, of course. So here I want N, A, and B. All right. And this is the Tellurium code, cell that I have. So this is what Tellurium is giving me. A starts at 1, so A is that blue curve, and B is the orange curve, and they both tend to 5 according to this equation that I did here. They both tend to 5. Does it make sense? I'm basically trying to do this guy here. I'm doing this guy here. A goes to B, B goes back to A. Does this result make sense? Why is it tending to 5? Why are they going to 5? Right, because of this. Yeah. So if K1 is equal to K2, you have A equals B in the long term, right? So generally for formality reasons, when we do this, you have your equation, and then when you do it equal to 0, we tend to put a star, saying that is the steady state. What I told him, what I told Jen, Niraj, was that A star is B star, like this, in the long term. Yeah. Star means the steady state. Because K1 is equal to K2. What happens if I do K2 equals 2? What will happen?
They would not converge.
They will not converge?
Yeah. I'm like, basically, one would have higher concentration, another would have lower concentration.
Which one? Which one
Which
will have the higher concentration? It's right there in front of you, John.
Yeah, I know. So I know you answer.
Go ahead, answer.
B would have higher concentration.
Okay, yeah, that's it. Correct. Well, Niraj?
B would have higher concentration.
Incorrect. A will have the higher concentration, I hope. Maybe I'm wrong. Yeah, A, the blue curve, A. Is it the blue? Yeah, it is because A is starting at 1. Yeah. So A is gets the higher concentration.
Hmm.
What else? What if I start with B equals 1? Will the steady state be any different if I start with B equals 1?
It should be the same, but the final value.
Okay, Jen said that the final values will be different. Do you agree with that, Niraj?
Yes, they would be different.
They will be different. What is constant? The ratio. So one goes to 1.4, the other is about 0.7. So you have still the same ratio of 2, but different final values. Perfect. So what Jen did before the correct attempt was A going to be at a rate no. So what I will do here what Jen did initially. So initially he did this. A goes to nothing at a rate k1 times A, and he did B goes to nothing at a rate k2 times A. So if you erase this, which is what you had before, you basically are losing A independently of B and losing B independently of A.
Actually, I meant A will generate B at the speed of.
Yeah, but what you meant was not in the equation. Okay, so your equations were in the Tellurium code, they are this. So what will this be? How will this behave? They both decay to 0 and nothing special. Make sense? So this would be the final output. Very different. So you guys will do that. So let me correct this. So take 5 minutes to implement it in Tellurium, like this, in your Colab file. And if you want, you can implement another one.
Spell this
Tellurium. I pasted on the chat. Do you have access to the Zoom? Ah, okay, okay. Yeah. So you're right in this. Oh, okay. Nice. Okay, perfect. So last class, James taught it to run locally. Is that right? James taught you guys to run Tellurium locally.
No, I just know how to run.
Okay, fine. That's perfect. And where is it? And if you want to be, no, yeah, you can start with that one. A goes to B, B goes to A, but then you will try the another one. Like, I don't know. Whatever, another one, I don't know. Yeah, we'll after that you will try the A plus C goes to B. That's the other one we'll try. But then setting in different initial concentrations of A and C. There is a very interesting effect that generally people study that in chemistry in high school when A and C start with different concentrations, but we call it the limiting reactant. but it's also cool to see it on the simulation. So I'll give you 15 minutes to implement what I have, and then try the A plus C going to B.
Yes.
Did you try? What are you plotting in your graph right there?
Sorry.
What are you plotting in the graph?
This only A plus C gives you B.
A and B?
No, I'm like I completed the one A plus C gives you B, right?
Yeah, but in the plot. So what is the
Right.
red curve
Oh, okay.
and what is the blue? I mean what I mean red is A or C, but are you showing both?
Yeah. So the A and C have taken concentration as one, so they both overlap.
Mm,
So the B is the orange curve,
okay.
and A and C both are overlapping.
So try A different than C, the initial concentrations. Ah, look at that. With A and C with initial concentrations different. Does that make sense?
Yeah.
My question here is why don't you end with C equals 1? Why C is going to 0.5 when one of your reactants started at 1? Let me try that. Because I think it should, it should go to 1. So A plus C goes to B. And the rate is K1 times A times C. Let's say A starts at 1, C starts at 2, B starts at 0. I don't have K2 here. And then I want to see all 3. Yeah, this is more like it. This is what I thought should happen. So if you start with one of your reactants at 1, your product should go to 1. Right?
Oh yeah, right. So I basically thought that we also need to define. I just like wrote for the condition of A and C also in new line defining like there should be minus K1 AC. So that's why I was getting different answers somehow.
Oh, okay, interesting. Yeah, no. Here is a one-way reaction.
So okay, so it assume everything by itself for the condition of A and
Yeah,
C also, right? Okay, cool.
yeah. So Tellurium handles this reaction very well. I mean, it transforms them into 3 ODEs and solve it. So Tellurium, I mean, I implemented this little reaction in 5 seconds. I will take probably 5 minutes if I had to write all these. Okay, so let's go back to the slides. Did you were you able to implement it on Tellurium, Jen? Could you implement it on Tellurium?
Uh,
Nice. Perfect.
Can we not add legend to the plot?
Yeah, you can add legend to the plot. Let me see if I have that. I mean, I surely have it on one of these plots. Um, huh, or I think it's this one. No. Uh, it is possible, but I don't remember
Okay.
to add the legends. Let me try. It is here somewhere, so it should give me the legend option when I do this. It is labels. Default is known, and yeah, so it's labels probably equals, and then all of
Oh yeah,
those, I
I was
guess.
able. Yeah, got it.
Yeah, that's it.
Hmm.
All right, let's keep going. Because if we end the class sooner, better, right? So about our sigmoid functions. This is what we're going to see later, but I decided to put a slide here because this is how you're going to, in the future, write your sigmoids in reactions. So they are called Hill equations, and they have the Hill exponent, this n. The n regulates how sharp the transition is, and this k here regulates where is the halfway between the 0 and the 1. So by regulating k, you're basically shifting your sigmoid left and right. But that's for the future. Okay, another exercise. Look how good it is. So you will choose one topic: protein degradation, protein synthesis, RNA transcription, or nuclear transport. And you will do 1, 2, and 3. Represent it chemically in a chemical reaction, represent as an ODE, and write it in Tellurium. It doesn't need to run in Tellurium. I mean, if you fail to code it and make it run, that's fine. But at least represent it as a diagram and build the ODEs for it of one of these topics. But I don't know anything about protein degradation. So go to Wikipedia and take a look at how it happens more or less and build a very simple as possible model about it with at least 2 nodes, at least 2 players. So choose one of them, the one you feel most comfortable or the one you are most interested in, and we'll see how what you got. Niraj, can you like describe your model? That's just one node, Niraj. 2 nodes minimum. How is a protein degraded in the cell? No, when a protein is degraded, it turns it into the amino acids. It is cleaved. That's not something you want to model, like explicitly the amino acids. But if you go to, maybe I should do that then. If you come here and you say protein degradation, and let's go to Wikipedia. Protein degradation. Proteolysis. Let's take a look at this. Intracellular protein degradation. Yeah, that's much more complicated than we need. So let's take a look at a diagram. Protein degradation diagram. And here we find this one. So here we have our substrate. Our substrate is connected to an E3, and via an E2, it gets an ubiquitin. And if it has an ubiquitin, it is then degraded via the proteasome. So we can make a, but of course, let's say we want 2 nodes, okay? Let's make something very simple with just 2 nodes. I have my substrate. My substrate becomes nothing, so it's degraded. And the E2 ubiquitin ligase is promoting this process. Does this make sense to you, Niraj?
Yep.
Based on this diagram here from Nature?
Yeah.
You can make it with more nodes, considering, for example, the proteasome as a catalyst. You can also consider E3 as another catalyst. You can consider multiple rounds of ubiquitination. So let's consider many rounds of ubiquitination. So I have S1 going to, I have S going to S1, promoted by our E, and then it goes to S2. Let's say it needs 3 rounds of ubiquitination. Sorry, this is 3. Then we will do this right here. If it needs 3 ubiquitins, like in the diagram, right? So then
Yeah.
our S1 is representing our protein with 1 ubiquitin, two ubiquitins, so on and so forth. Does this make sense?
Yes. No, but can I not consider, let's assume I have a dimer protein, which basically breaks into two proteins. So A gives 2B. Simple.
But that would not be protein degradation. That would be a very specific cleavage of a protein or a complex breaking down into smaller into the components of the complex. That this is not,
Okay.
it's not protein degradation. Protein degradation is the digestion, the full digestion of the protein inside the cell into amino acids.
Right.
So you would have, like, instead of A going to 2B, you would have, I don't know how many amino acids your protein has, 1,000. You would have A going to 1,000B.
Right.
And then you have to know explicitly how many amino acids your protein has.
But again, there would be a question like, are all amino acids similar? There might be different amino acids in that
Yeah,
also.
yeah.
So yeah.
So um it's it's always a question of, do you care about those amino acids? Like, what are you trying to model? You know?
Hmm.
Sometimes you don't care about those amino acids. What are they? You just care about, you have the protein or not. That's what you want to know. If that protein is high, the cell will die. So you just care about whether or not the protein is there or not. If it's being
Right.
degraded or not. Like, at what rate it's being degraded. This rate depends on what? How can you manipulate the rate of degradation of this protein? That's what you care.
Hmm.
What about you, Chen?
So, I'm simulating the generation of RNA.
Okay, okay.
So,
Let's take a look.
this is definitely.
What is the
So, the R means RNA, D means DNA,
Okay.
G means protein. So, the RNA comes from DNA, but DNA is always there. It's not consumed. So, R is generated from nothing. And the DNA, RNA can generate DNA.
No.
Like, it's a reverse process.
It does no. So the this is an important, yeah, and this is so let me include the other guys in the discussion. So the structure of Chen's model, so Chen is modeling RNA replication. Sorry, not an RNA replication. He is modeling the dogma, the central dogma of biology, which is DNA generates the RNA, and then RNA generates protein. So, and understanding biology is critical to model it correctly, because there is a, so from DNA to RNA, the molecule that does this is the RNA polymerase. Okay. And the RNA polymerase just transcribes DNA into RNA. The generation of another DNA molecule, so from DNA to DNA, that's done by the DNA polymerase. It's another molecule, another complex altogether. So this is the process that happens when the cell divides, goes through the cycle, the cell cycle.
Some virus RNA can generate. RNA virus.
They, no. So they don't generate DNA, but it doesn't mean they don't have a genome. So their RNA functions as a DNA. But this process is not reversible.
We cannot use RNA as a template to generate.
No. Not as far as I know. But the rest of your model is fine. The rest of your model is completely fine. So you would have like a DNA that is on or off. If it's on, you have a certain rate of production of RNA. You also have RNA decay, so it tends to a certain value, which is perfect. And then depending on the level of RNA you have a production of protein, because RNA is promoting the production of protein. Is that right? Is that your structure?
Yeah.
Yeah, which is perfect. So basically what you have is you have nothing going to RNA, and whether or not you have DNA, it promotes the production of RNA. And whether or not you have RNA, it promotes the production of your protein. Right? The sigmoidal function? Okay, no problem. And then this guy also decays, and this guy also decays, so you don't have those infinites.
Yes.
That's a reason. This is a reasonable model. You know, it's a very simple two nodes model, and DNA could be a binary stuff, 0 or 1, because it can be on or off depending on whether you have that piece of DNA activated or not. Fine. Okay, that was good. That was very good. And nuclear transport is a very interesting one, nuclear transport. Let's take a look at some diagrams. This one is a good one. So the nucleus has these nuclear porous complex. NPC, it stands for nuclear porous complex. Is that it? Nuclear pore complex. It's a passage, but it's a passage that just allows proteins to pass that are bound to very specific proteins called importance. And it just allows export of proteins if they are connected to the exporting proteins. So this nuclear pore complex is an intelligent system, that allows or not the transport of certain proteins, whether or not, depending on whether they are connected to these importings or exportings. And these importings and exportings, this is very interesting, they just bind to proteins that start with a certain sequence of amino acids. So when the protein is translated and it needs to go back to the nucleus, back inside the nucleus, it's already written in the DNA that this protein has to be targeted to the nucleus. And if a certain protein or RNA molecule needs to go out of the nucleus, it's already encoded in the DNA whether this protein or RNA molecule has to go out or not. So the DNA even tells where the protein has to go, which is very interesting. And here we have, for example, if we were to model only the import, we can have, for example, let me do this quickly here just for curiosity. So what I will do is I have protein. I don't care whether it's being produced, whether it's being destroyed. I just have a protein, and this protein is in the cytoplasm. And this protein can enter the nucleus. So I have another node that is the same protein in the nucleus. So I have index C for this protein in the cytoplasm, and I have this index N for the protein in the nucleus. So now the index means the compartment of the cell where the protein is, but it doesn't mean that the protein is any different. But how I want to do this is not actually a direct flux. I want to connect this guy with my importing, and then I will have my protein in the cytoplasm complex with my importing protein. Now I have a complex and I'm calling it PCI. Then this guy enters the nucleus. Shit. Let me close this. So for some reason, the sound from my PC transmits to the TV, but from Zoom, specifically from Zoom, it doesn't get transmitted to the PC, to the TV, sorry. And then after connecting to the importing, I will have my protein in the nucleus. And I can assume that once it enters, it disconnects from the importing, but that's not as trivial as that. So simple ways to implement these very complex systems, and you can make it as sophisticated as you like. And I think today we learned the basic tools to do that. So when you see a diagram like that, or a huge diagram, you already can imagine the reaction and know how to implement them in Tellurium and write the ODEs. For example, in your case, if you want to not use Tellurium, if you want to use another software, something that you can optimize better, you need to know how to write the ODEs, because the ODE is the ultimate representation of what is going on here. So I hope the objective of the class was achieved, which is precisely that, being able to look at a diagram like this and represent it as a kinetic reaction, implementing it in Tellurium, and writing the ODs. Finally, this is the first homework. This homework was my idea. And it's a paper about a real case of someone that invented a network. It's a very simple network to represent a oscillatory behavior in cells. And it's a short paper, it's 4 pages. And the only thing you need to do is answer these questions reading the paper. So hopefully you can do that quickly, and no more than 1 page, because I don't want you guys to freak out. It has to be something you solve in like 20 to 30 minutes. It might take more if you are not familiar with the language, but if you're not familiar with the language, you can say in the homework, this is what I understood, blah, 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 blah. And then here in next class, we will discuss this paper. This paper is not free access, but IU grants you access to the paper. If you don't know how to do that, ask us and we will help you accessing the paper. Besides that, from now on, we'll start discussing about your project ideas. I mean, from this class forward, because since we have only 2 of you, we want to narrow the class to your personal interests. Otherwise, we'll have to like the full course for many people is about like motifs, basically motifs, and studying the motifs. So we do parameter scan, we do sensitivity analysis, we do phase plots, we do stability analysis, but that might not be your interest for your project. So we can tune the classes towards your goals in this class. So next class, bring me some idea of what you expect from the class and what project you want to build in this class. Like, what is your idea? For example, you said you want to optimize something, or Niraj said he's building Boolean networks. So perhaps Niraj would think, for example, let's take these Boolean network I have and let's make it kinetic. Let's make it not Boolean and see what happens and compare. Or no, I want to stick with the Boolean and I want an approach other than Tellurium that helps me out to solve my project. So bring us an outline of that so we can figure out what classes we'll give next. Okay? And that's all from today. You guys are free to go. So hopefully see you next week. If you plan on attending, just show up. If you plan on dropping the class, send us an email so we can decide whether the course continues or not. It's always good to know the logistics. So anything from you, Niraj?
Just wanted to know what all are we planning to cover in this course?
So the things I said, so we learn basic motifs, reaction motifs, which is simple model structures that you see repeatedly on models. Then using Tellurium, learning how to build phase plots, do parameter scanning, stability analysis, do like null line stuff. I don't know if you guys heard of that, but
Yeah.
it's basically a dynamical systems class focused on the first order differential equations for reactions. And of course, using them to model biological scenarios. That's how the course is originally designed.